Thank you for visiting my video again. Today I'm talking about another key point of this paper, a quantitative model of nitrogen fixation in the presence of ammonium. I'm the first author of this paper, Keisuke Inomura, and this paper was published in 2018 in PLoS ONE. So the main point today is the figure 7. To provide a brief background, nitrogen-fixing organisms need respiratory protection, which is high respiration to protect nitrogenase from oxygen because respiration consumes intracellular oxygen. So generally, it has been thought that respiratory protection is mainly to protect nitrogenase or nitrogen-fixing enzyme, but it has not been clear when this respiratory protection is happening. And it's natural to assume that it only happens when nitrogen fixation is happening because that's basically for that. But this result shows that even if nitrogen fixation is not happening, high respiration or respiratory protection seems to be happening. I'll explain that with panel 2 because these two panels are really similar. And dots are data, and the solid line is simulation, and dotted line is simulation 2. So the difference between these simulations are simulation 1 is the case with respiratory protection even when nitrogen fixation is not happening, and simulation 2 is the case with respiratory protection only when nitrogen fixation is happening, or the domain C. So by comparing to the data, we find that the simulation 1 is better representative because the data increases respiration rate with C to N ratio, which is what's happening in simulation 1. Because the increase can be explained by respiratory protection. As C to N ratio increases, more excess carbon is available, which is channeled into respiratory protection. So, but simulation 2 doesn't capture that because the trigger is only nitrogen fixation. So if simulation 1 is similar to data, that suggests that respiratory protection is happening even when nitrogen fixation is not present. So why do they do it? Why do cells turn on respiratory protection even when nitrogen fixation is not happening? It sounds unnecessary, but we thought about a few hypotheses. One is simply cells cannot turn off respiratory protection. It's hardwired to the cell, and there is no intracellular control over the level of the respiratory protection. As long as carbon comes into the cell, it just consumes as much as they can. And the second hypothesis is that they do it intentionally to protect nitrogenase even when nitrogen fixation is not happening. Nitrogenase must be kept in low oxygen environment, and respiratory protection certainly helps preserving it. And the advantage of that may be in a dynamic environment, when suddenly oxygen concentration decreases or carbon concentration increases, enabling nitrogen fixation. And if there is a preserved nitrogenase, cells can instantly start fixing nitrogen. So that means instant growth. And in the competitive environment, it may be useful. So these are the hypotheses. We are not still clear about it, but what's clear about this study is that the respiratory protection is happening even when nitrogen fixation is not happening.